So good day everyone. So we're here today to discuss our assigned topic, the molecular basis of inheritance. And we are your presenter for today. I am Alden Matillas and with me is Nacario Charlene, Turin Costales, Robin Rosby, Gian Atayan, Velasquez Herrera. So to start, let's discuss the contribution of these four, four individuals to the discovery of DNA or the three dogma. So to start, Rosby, proceed.
In 1962 Nobel Prize in Physiology and Medicine, Watson, James, and Francis Crick discovered the molecular structure of nucleic acid and the structure of deoxyribose nucleic acid. What about Rosalinda Franklin? So ni Rosalinda Franklin discover na ti DNA in 1953. Next slide.
Um,
The structure of DNA. DNA is composed of 4 nucleic acids, each contain of adenine, cytosine, thymine, and guanine. The amount of adenine is equal to thymine and guanine is equal to cytosine, and purines is equal to pyrimidine and in Chargaff rule. So ni purines contains so na ti guanine ka ni adenine. Tas ni pyrimidine ka cytosine kini thymine. Tas numapancho ti RNA ka adati uracil. Tapos, DNA is a double-stranded helix with antiparallel strand by Watson and Crick. The nucleotides in each strand are linked by 5' to 3' phosphodiester band. The base on the opposite strand are linked to the hydrogen bond. Adenine is with thymine and guanine with cytosine. Tas lumapan mo ti RNA ni thymine masuktan ti uracil. Tas in illustration, makikita natin yung mga structure ng thymine, adenine, cytosine, kani guanine. Algibangi ka tayo sugar, phosphate, carbon. Okay? Backbone. Ito A, ati structure ti DNA aja anti-parallel. Tas ati J unat na.
So, ano ba yung sinasabi na anti-parallel ni Rosby?
Yeah, the A. Anti-parallel meaning aji agkakasuda ji in a different direction, opposite. Aji may isa ka mapanti 5' to 3' tas may jibangir ka 3' to 5'.
So, ito yung 5' to 3'. Then, yung anti-parallel na sinasabi dito yung 5. Dito naman yung 5. Then, start dito sa 5. Then, to 3'. So, complementary siya. So, next, Ms. Rosby.
Next slide. Tas, ito bangir aji mas visual na kuana. Nga dati minor groove kini major groove. Minor groove aji bas-basit na gap da. Tas ni major aji dadakal na gap. So you have the base, oxygen, hydrogen, carbon, sugar, phosphate, backbone, and the phosphorus. The basic principle to template strand. The relationship between structure and function is manifest in the double helix since the two strands of DNA are complementary. Each strand acts as the template of the building of a new strand in replication. DNA replication. A parent molecule unwind, the two new daughter strands are built based on base pairing rule. So, ati tibabak aji general process na. The parent molecule has two complementary strands of DNA with each base paired hydrogen bond with a specific partner A with T and G with C. So, ati J, magpakatatay jing mo na nga, slides. And then B, The first step in replication is the separation of DNA strand. Then C, each parental strand now serve a template that determine in order of nucleides along with a new complementary strand. Then D, the nucleotides are connected from sugar phosphate backbone of the new strand. Each daughter DNA molecule consists of one parent strand and one new strand. DNA replication is semi-conservative. Each strand daughter molecule is one half new and one original strand was used template to make new strand. So meaning semi-conservative meaning is contains of new strand and the old strand. Tapos makikita natin yung original strand, yung replication fork, replication bubble, and replication. So, kasla ATG summary T replication. Strand, original strand, and then the replication bubble. So, nag-start yung replication.
So, bali sinipay lang nito na hindi lang isa or di lang sa isa nag-isa yung ay nagmumula yung replication bubble kundi multiple siya. Dito. Isa, dalawa, tatlo. So, hindi lang isa nagkukan ng replication bubbles. So, proceed to the DNA replication.
DNA replication. The copy of DNA is remarkable in its speed and accuracy, involving unwinding of bubble helix and synthesizing new strand. More than dozen enzyme and other proteins participate in DNA replication. The replication of DNA molecules begin at a special site called origin of replication, where the two strands are separate. So, in that image, dito di mangrugrugyan ti pag-replicate ti DNA. Origin of replication. Or eukaryotic chromosome may have hundreds or even thousands of replication and origin. So, ibagbaga na dito is illustration na di kasanati replication bubble. Replication began at a specific site where the two parent strands separate and form replication bubbles. The bubbles expand literally as DNA replication proceeds in both directions. And then eventually the replication bubbles fuse and synthesis of daughter strand is complete. So makita tayo illustration nga ti bubble ko. So ti pagragagyanan na. And then multiple have the replication fork, template strand, kanid new daughter strand. Then maka-form dan to T2 daughter DNA molecule. Thus in image 1, in eukaryotes, DNA replicates begin at many sites along the giant DNA molecule of each chromosome. Thus in B, the micrograph, the replication bubbles are visible in the DNA of a cultured Chinese hamster cell. Ito dong, hindi ko may todo yung.
So, ito yung one. Ito ba yung tinatawag na replication bubbles?
Ito yung replication bubbles. Lagi dyan, buo. Kita.
So, let's move with the mechanism of DNA replication. DNA replication is catalyzed by DNA polymerase, which needs an RNA primer. RNA primerase synthesis primer on DNA strand. DNA polymerase adds a nucleotides to the 3' end of the growing strand. So, this image illustrates the single-stranded region of the parental DNA. Ito siya. Then, ito naman yung primase. Then, yung replication niya nag-start sa 5' to 3'. Primase join the RNA nucleotides into primer. Then DNA polymerase adds DNA nucleotides to primer. Then ito na siya. Ito yung RNA primer, then ito naman yung newly made DNA. So ito naman yung DNA polymerase. Bali, general illustration lang to. Then we will discuss more in the next slide. DNA synthesis on the leading strand is continuous. The lagging strand grows the same general direction as the leading strand, in the same direction as the replication part. However, DNA is made into the 3' to 5, 5' to 3' direction. Therefore, DNA synthesis on the lagging strand is discontinuous. So, ito yung key term niya. Pag lagging strand, discontinuous din. Pag leading strand naman, continuous siya. DNA is needed as short fragments, or also known as the Okazaki fragment, that are subsequent ligated together. Ito yung tinatawag na leading strand. Then, ito naman yung lagging strand. Ito naman yung lagging strand template. Leading strand template naman ito. Then, ito yung Okazaki fragment na tinatawag. Then, parental DNA. Then, ito yung mga enzymes na madidiscuss mamaya. So, in this image shows the DNA, parental DNA, ito siya. Then, DNA polymerase, ito naman siya. Then, the Okazaki fragment. So, tinatawag discontinuous, so nagkakaroon siya ng fragment o may butal-butal ba. Ito naman yung lagging strand. Leading strand. Then, as part ito yung overall direction ng replication. Then next stage, DNA polymerase 1 degrades the RNA primers and replace it with DNA. Newest DNA, ito siya. Then, ito naman yung DNA polymerase 1 na magde-degrade ng RNA primers and replace it in DNA. This daughter strand is now completed. So, many protein assist in DNA replication. DNA helicase unwind the double helix. The template strand are stabilized by other proteins. Single-stranded DNA binding proteins make the template available. RNA primase catalyzes the synthesis of short RNA primers to which nucleotides are added. DNA polymerase 3 extend the strand in the 5' to 3' direction. DNA polymerase 1 degrades the RNA primers and replaces it with DNA. DNA ligase joins the DNA fragments into continuous daughter strand. So in this image, ito siya. Lagging strand, RNA primer, RNA primase, RNA primer naman yung kasalun yan. Then, DNA polymerase 3, ito siya. Then, DNA polymerase 1. So, see the difference color, magkaiba siya ng color kasi magkaiba sila ng function. Magkaiba yung function ng DNA polymerase 1 sa DNA polymerase 3. To be discussed later on. So, DNA ligase, ito naman. Then, ito yung mga enzyme na involved sa DNA replication. Si helicase, function niya to unwind the parental double helix. Then si binding protein naman, stabilize the separate strand para hindi na siya magdikit sa mga strand. Kumbaga siya yung magsusupport sa na-separate strand. Si primase adds a short primers to template the strand. Then DNA polymerase 3 binds the nucleotide to form new strand. Then DNA polymerase 1 removes, or also known the extra nuclease, remove the RNA polymerase and insert the correct bases. Ito yung nagkakaiba. Ito sinatawag ko kanina. Ligase then the Okazaki fragment. Fragment and seals other meat in sugar phosphate backbone. Replication. Helicase protein binds the DNA sequences called the origins and unwind the DNA strand binding proteins to prevent single strand from rewinding. Primase protein make a short segment of RNA complementary to the DNA primer. So ito siya. Ito yung tinatawag na helicase na mag-a-unwind ng strand, DNA sequence. Saka ito naman si binding protein. Then, ito naman si primase protein natin. DNA polymerase enzyme adds a DNA nucleotides to RNA primers. Ito na siya. Nag-add na siya ng nucleotides to the RNA primers. So, tingnan nyo yung start from 5' to 3' siya. Then sa taas naman, 5' to 3' din. DNA polymerase proofread bases added and replaced the incorrect nucleotide. So, si DNA polymerase may capable siya na proofreads, kumbaga nakikita na yung mga mistake para ma-prevent din. Papalitan niya ng specific nucleotides o kumbaga yung mga correct bases niya. Leading strand synthesis na continuous in a 5' to 3' direction. So, ito siya. This continuous synthesis produce 5' to 3' DNA segment called Okazaki fragment. Ito yung tinatawag na Okazaki fragment. Sa baba. Leading strand synthesis continues in a 5' to 3' direction. Ito naman yung leading strand. Kumbaga wala siyang butal-butal o wala siyang fragments. This continuous synthesis produce 5' to 3' DNA segments called Okazaki fragment. Ito naman siya, yung Okazaki fragment. Nagkakaroon ng gap. Exonuclease activity of DNA polymerase 1 removes the RNA. Ito yung nagre-remove ng mga RNA primers naman, si DNA polymerase 1. Polymerase activity of DNA polymerase 1 fills the gap. Ligase form the bonds between the sugar phosphate backbone. So ito naman si ligase naman na nagkukon sa mga gap ni Okazaki fragments. Then dito, replication part overview. Enzyme unwind the parental double helix. Process 2. Same process, protein stabilize the unwound parental DNA. Process 3. The leading strand is synthesized continuously by DNA polymerase. Then, for the lagging strand is synthesized by discontinuously RNA polymerase synthesize a short RNA primer, which is then extended by DNA polymerase. So move to the key process. DNA polymerase digests the RNA primer and replace it with DNA. Then the last process, DNA ligase joins the discontinuous fragment of the lagging strand. So ito yung kabuuang process ng replication. Proofreading. DNA must be faithfully replicated, but mistake occurs. DNA polymerase or DNA pool insert the wrong nucleotide base in 1 per 10,000 bases. DNA pool has a proofreading capability and can correct the errors. Mismatch repair, wrong inserted base can be removed. Excision repair, DNA may damage by the chemical radiation, etc. Mechanism to cut out and replace with the bases. So yung proofread, bali niya, binabasa niya yung code kung tama ba, then nire-replace niya into correct bases. Then mutation. A mismatching of base pair can occur at a rate of 1 per 10,000 bases. DNA polymerase proofread and repair accidental mismatch pair. Chances of mutation occur at any one gene is over 1 in 100,000. Because human genome is so large, even at this rate, mutation add up, each of probable inherited 3 to 4 mutation. Proofreading and repairing DNA. DNA polymerases proofread newly made DNA, replacing any incorrect nucleotides. In mismatch repair of DNA, repair enzyme correct errors in base pairing. In nucleotide excision, DNA repair, nuclease, cut out, and replace damaged stretches DNA. So, ito naman yung kanya process niya. A timing dimmer disturbs the DNA molecules. A nuclease enzyme cuts the damaged DNA strand at 2 points and the damaged section is removed. So, ito yung nuclease. Third process, repair synthesis by the DNA polymerase fill in the missing nucleotides. So, na-fill na yung missing nucleotides niya. Then, last process, DNA ligase seals the free end of the new DNA to the old DNA, making the strand complete. So, siya yung nag-pitch or nag-seal ng mga gaps nila. Accuracy of DNA replication. The chromosome of E. coli bacteria contains of 5 million bases pair, capable of copying this DNA in less than an hour. The 46 chromosome of human cell contain about 6 million base pair of DNA. Takes a cell few hours of copy this DNA with amazing accuracy, an average of 1 per billion nucleotides. So guys, kumbaga kung nagkakasakit tayo, di ba, prescribe ng doktor na magkaroon tayo ng antibiotic. So, ano kaya yung purpose ng antibiotic na yan sa tingin niyo?
Tapno ma-cure na yung sakit.
Ano pa yung other reasons? Okay, given na para ma-cure yung disease or yung illnesses.
Kaya kasi
Ang kwan, may mga ibang antibiotic na inhibit yung replication ng mga bacteria or yung mga viruses. Kaya yung mga antibiotic minsan, prescribe ng doctors natin para ma-prevent yung replication ng mga bacteria sa katawan natin to preventing illness worsen. Kumbaga, hindi na lumala yung sakit. Kaya ito yung mga purpose ng mga antibiotic. Hindi lang sa nakagamot siya. Given na nakagamot siya, pero yung main purpose ng antibiotic na ilan, di ko lang sure yung mga brand niya or yung specific na antibiotic na yan. Nagkukan din siya ng kinokontra niya yung replication ng mga bacteria sa katawan natin para hindi na lumala yung sakit. Then, imagine nyo yung yung E. coli contains about 5 million bases pair capable of copying this DNA less than an hour. So, ibig sabihin, mabilis yung replication niya. Kumbaga, mabilis din yung mag-worse yung sakit mo. Kumbaga, lumala yung sakit mo. Then, next, protein synthesis. The information content of DNA is the form of a specific sequence of nucleotides along the DNA strand. The DNA inherited by the organisms lead the specific trait by dictating the synthesis of protein. The process by which DNA direct protein synthesis genes expression includes two stages called the transcription and translation. Then now we will move to the transcription and translation. So tapos na yung replication, then next process niya transcription, and this transcription process will be discussed by Chuyin, Costales, and Macario Chardi. So, proceed.
Transcription and translation. Cells are governed by a cellular chain of command into DNA to RNA and protein. So transcription is the synthesis of RNA under the direction of DNA. Produces messenger RNA or mRNA. Translation is the actual synthesis of polypeptide which occurs under the direction of mRNA and occurs on ribosomes. So transcription begins when an enzyme called RNA polymerase attaches to a DNA, a segment of DNA, and that is the gene. So a gene contains the code to build a specific protein which is a macromolecule and is made up of a sequence of amino acids in a specific order. In a eukaryotic cell, the nuclear envelope separates transcription from translation. Extensive RNA processing occurs in the nucleus.
So, ito naman yung image na ni-illustrate niya yung process ng transcription.
Oo, ng
Then,
transcription.
from DNA to pre-RNA to mRNA na galing siya sa nucleus, then after ma-provide ng or maka-release ng product na RNA, lalabas siya, then pupunta siya sa ribosomes, which is the protein synthesis. Then, dito siya magkakaroon ng specific amino acids or yung tinatawag kanina ni, ni Chewing na specific proteins.
Eukaryotic cell. The nucleus provides a separate compartment for transcription. The original RNA transcript, called pre-mRNA, is processed in various ways before leaving the nucleus as mRNA. Transcription is the DNA-directed synthesis of RNA. RNA synthesis is catalyzed by RNA polymerase, which pries the DNA strands apart and hooks together the RNA nucleotides, follows the same base pairing rules as DNA, except that in RNA, uracil substitutes for thymine.
So, ito na yung sinasabi kanina ni Miss Robino na substitution. Sa DNA, thymine siya. Pero pagdating sa RNA, yung substitute naman ni thymine is uracil. So, dito siya. In this illustration, makikita natin na from guanine to cytosine, adenine to uracil. Kumbaga sa DNA yan, adenine to thymine siya. Pero sa RNA, napalitan siya ng uracil.
So, yeah, RNA will never contain thymine. So, whenever RNA polymerase is adenine, it always pair with uracil.
Yun.
RNA is single-stranded, not double-stranded like DNA. So, RNA is short, only one gene long, where DNA is very long and contains many genes. RNA uses the sugar ribose instead of deoxyribose in DNA. RNA uses the base pair, the base uracil instead of thymine in DNA. There are 5 types of RNA. The messenger RNA carries information specifying amino acid sequences of proteins from DNA to ribosomes. And second one is transfer RNA or tRNA. Serves as adapter molecule in protein synthesis. Translates mRNA codons into amino acids. And the third one is ribosomal RNA. Plays catalytic or ribosome roles and structural roles in ribosomes. And the fourth one is primary transcript. Serves as a precursor to mRNA, tRNA or the ribosome RNA or tRNA. Before being processed by splicing or cleavage, some intron RNA acts as a ribosome, catalyzing its own splicing. And the last one is small nuclear RNA. Plays structural and catalytic roles in spliceosomes, the complexes of protein and RNA that splice pre-mRNA.
So, madagdag lang Choy, pagdating dito sa part na to, baling na compare na yung comparison ni RNA sa DNA. So, tama ba? So, pag dito, double CRNA, single-stranded lang siya. Then si DNA naman, double-stranded or double helix. Yep. Then,
Yun din.
one gene long or DNA long contains many genes. Uses, o dito na, yung sugar na nagagamit sa RNA is ribose. Sa DNA naman, deoxyribose. So, pwede naman sa nucleotides na nagagamit is sa RNA, uracil naman siya. Then sa thymine, sa DNA naman is thymine. So, ano kayang yung ano kaya yung parehas sila na included. Kumbaga, mayroon silang similarities. Kasi na-discuss na dito yung comparison nila, yung pinagkaiba ng mga components nila. Then, doon naman tayo sa similarities nila. Ano yung nandun sa kanila na parehas na naggamit sa process?
The genetic code, ay, nucleotides. Yun.
Yep, yun. Sa nucleotides, yung tatlo.
Ah,
Di ba
yung
may palitan lang ng dalawa? Isa.
cytosine, guanine, adenine.
Yun. Yun yung similarities naman nila. Then next.
So, synthesis of an RNA transcript. There are 3 stages of transcription: initiation, elongation, and termination.
Yung promoter kasi siya yung kumbaga, anong tawag din? Siya yung tumutulong na magkwan ng strand.
Initiation. After RNA polymerase binds to the promoter, the DNA strands unwind and the polymerase initiates RNA synthesis at the start point on the template strand. So, RNA polymerase, RNA polymerase causes a particular area in the DNA helix to unwind and separate the DNA into 2 strands. Template strand and the other one is the coding strand. The template strand is transcribed by the
Ay, Choy, nawawala yung signal mo. So, pagdating naman sa elongation, the polymerase move downstream, unwinding the DNA, elongating the RNA transcript from 5' to 3'. In the wake of transcription, DNA strand deform the double helix. So, ito yung sinasabi ni Choy.
in the elongation, using the template strand, RNA polymerase uses the base pair rule to assemble the nucleotides in the nucleus into a complementary strand of an RNA. And the last one is the termination. Eventually, the RNA transcript is released and the polymerase detaches from the DNA. When transcription is complete, the mRNA which is small enough to fit in the nucleus.
Synthesis of RNA transcribe initiation. Promoters signals the initiation of RNA. Ito na yung sinasabi ko kanina. Ito yung mag-signal sa initiation ng RNA synthesis. Then, transcription factor help the eukaryotic RNA polymerase recognize the promoter sequence. So, Choy, discuss mo na to. Dali wala ka kanina. First, yung eukaryotic promoters. Ito na yun. Then, several transcription. Ito yung tinatawag na promoters niya. Then, yung template strand. Then, ito yung starting point. Ito na last part ng synthesis. Okay? Synthesis of an RNA transcript elongation.
Okay, synthesis of an RNA transcript elongation. RNA polymerase synthesizes a single strand of RNA against the DNA template strand, the antisense strand, adding nucleotides to the 3 and 2 of the RNA chain. As RNA polymerase moves along the DNA, it continues to untwist the double helix, exposing about 10 to 20 DNA bases at a time for pairing with RNA nucleotides.
So, ito yung RNA polymerase na tinatawag kanina. Then, ito yung direction ng transcription niya downstream. So, pag ganun siya, from 3 prime, mag-end siya, siya 3 prime. From 5 prime to 3 prime. Then, continuous yung kanyang nucleotides niya, yung pag-insert ng nucleotides niya, RNA nucleotides niya. Then, ito naman yung template strand, then the non-template strand. Then, ito yung sinasabi ko kanina, lalabas na siya. Ito yung newly made RNA. Pagkaras ng transcription, lalabas na siya. Or the translation with the elongation process. So, next.
Next is the synthesis of RNA transcript termination. Specific step in the DNA termination. When one of these is by the base, the RNA transcript is released from the DNA and the zip up again. Here is the transcription overview from DNA to mRNA protein.
From DNA, moving nucleus to mRNA, then to the ribosome for the protein synthesis.
NMS. Ito yung medyo polymerase, ito yung nagbibilog. Diba? Iso ba?
Ayan.
Tapos,
Then, ato yung RNA nun, then ito yung RNA yung makuha ko, mga bubo, then natin may insert nga, RNA nucleotides. Then, after the process, the RNA synthesis, bali rumuha pa sunod sa di bagba ni Choy nga, termination.
terminator.
When the transcription is complete, the mRNA which is small enough to fit the nuclear pore, will take the genetic code out to the nucleus and ayun, pupunta sa ribosome.
Then next,
Next is the termination RNA. Most eukaryotic mRNA aren't ready to be transcribed directly after being transcribed from DNA. mRNA requires processing. Transcription of RNA processing occurs in the nucleus. After this, the messenger RNA moves to the cytoplasm for translation. The cell adds a protective cap to one end and a tail of ACE to the other end. This both function to protect the RNA from enzymes that would degrade. Most of the genome consists of non-coding regions called introns. Non-coding regions may have specific chromosomal functions or have regulatory. Magda ka lang.
when So, introns also allow the alternative RNA splicing.
Thus, a copy of is converted into messenger RNA by doing two things. First is to add protective bases to the ends. Second is to the introns.
So,
Next.
ito yung sinasabi ni Charlie. Then, pakibasa mo na, Charlie.
Addition of ends. Each of a pre-mRNA molecule is modified in a particular way. The 5
5' N receive a modified nucleotide path. The
and
3'
uh,
N get a Pauline A tail.
polymerase
So, ito yung ano yung mistake na transcription, then the DNA to pre R pre-mRNA to mRNA, matured mRNA, to
ribosome.
lumapit yung nuclear pool, then pupunta siya sa ribosomes for translation. Ito naman yung yung ammonified guanine nucleotides added to the 5' N. Ito siya. Ito yung
ATP5N
modified guanine. Ito yung nai-add. Guanine nucleotide. Modified guanine nucleotide. So, ito naman yung poly A tail na tinatawag. 52 to 50 adenine nucleotides added to the 3' N. So, ito naman yung part ng
protein coding.
protein coding segments niya. Then, ito yung polyadenylation signals niya. Start codon and stop codon. Kung saan sa mag-stop, then mag-stop siya.
Poly niya polyadenylation
Polyadenylation signals.
contain T3 UTR.
Yep. Then,
Next is the RNA splicing. The transcript from the DNA is called pre-mRNA. It contains transcripts of both introns and exons. The introns are removed by a process called splicing to produce messenger or
DNA then composed of exon, intron, exon. Then next, the initial mRNA transcription. Ito yung leader. Ito naman yung trailer. Then ito yung mga exon and introns niya. Then mRNA process. So mag-insert siya ng cap dito. Then dito naman yung poly A tails. Then ma-splice yung introns, splicing process. Matatanggal yung introns, then magdudugtong tong mga exon. Saka yung poly A tails niya. Then ito na yung matured na mRNA na product ng transcription.
Next is the RNA processing. Proteins often have a modular architecture consisting of these structural and functional regions called domain. In many cases, different exons code for the different domains in a protein. So that is the features.
Transcription process, RNA processing, translation. Ito yung mga tinatawag ni Charlene na 3 domain. First domain ito. Third domain, ito naman. At saka second domain. So yung first domain siya yung exon 1. Yung second domain siya exon 2. Saka yung third domain siya yung exon 3. Yung polypeptide. Next. Translation na siya. Then, this translation will be discussed by Romero, Velasquez, and Atayan, Gian.
RNA processing. Proteins often have modular architecture consisting of discrete structural and functional regions called domains. In many cases, different exons code for the different domains in a protein. Translation. Translation is the RNA-directed synthesis of a polypeptide. Translation involves the mRNA ribosomes or the ribosomal RNA, transfer RNA, and genetic coding codons. The genetic information is encoded as a sequence of base triplets, or codons. The genetic code codons tree-based code for the production of a specific amino acid sequence of three of the four different nucleotides. The genetic code for amino acid is determined by sequences of three nucleotides known as codons. Each codon corresponds to a specific amino acid. Here are the three examples of the amino acid is methionine or the start codon or the AUG, and the second one is phenylalanine codons or the UUU or the UUC. The last one is the serine codons is CU, UCC, UCA, and the UCG. Keep in mind that the genetic code is degenerate, meaning multiple codons can code for the same amino acid. Absolutely, on with four bases, the ATCG and three position in each codon, there are indeed is equal to the 64 possible codons. These codons code for the 20 standard amino acids and include three stop codons. Um, Transfer RNA or the tRNA molecules are generally depicted as having a clover-leaf structure with three lobes and anticodal lobe forms one arm of the L shape. These structures have tRNA carry specific amino acids to the ribosome during protein synthesis. Transfer RNA consists of a single RNA strand that is only about 80 nucleotides long. Each carries a specific amino acid on one end and has an anticodon on the other end. A special group of enzymes pairs up the proper tRNA molecules with their corresponding amino acids. tRNA brings the amino acids to the ribosomes. The anticodon is the three RNA bases that matches the three bases of the codon on the mRNA molecule. Ribosomes facilitate the specific coupling of tRNA anticodons with mRNA codons during protein synthesis. The two ribosomal subunits are constructed of proteins and RNA molecules named ribosomal RNA or the rRNA. Computer model of functioning ribosome. This is a model of a bacterial ribosome showing its oval shape. The eukaryotic ribosome is roughly similar. A ribosomal subunit is an aggregate of ribosomal RNA molecules. Schematic model with mRNA and tRNA. A tRNA fits into a binding site when its anticodon base pairs with an mRNA codon. The P site holds the tRNA attached to the growing polypeptide. The A site holds the tRNA carrying the next amino acid to be added to the polypeptide chain. Discharged tRNA leaves via the E site. Building a polypeptide, we can divide translation into three stages: initiation, elongation, and the termination. Initiation of translation. The initiation stage of translation brings together mRNA, tRNA bearing the first amino acid of the polypeptide and two subunits of a ribosome. A small ribosomal subunit binds to a molecule of mRNA. In a prokaryotic cell, the mRNA binding the site on this subunit recognizes a specific nucleotide sequence on the mRNA, just upstream of the start codon. Initiation tRNA with the anticodon or the UAC. Elongation of the polypeptide chain. In the elongation stage, amino acids are added one by one to the preceding amino acid. Codon recognition. The anticodon of an incoming antimonacil tRNA base pairs with the complementary mRNA codon in the A site. Hydrolysis of GTB increases the accuracy and efficiency of this step. Peptide bond formation. An RNA molecule of the large subunit catalyzes the formation of a peptide bond between the new amino acid in the A site and the carboxyl end of the growing polypeptide in the P site. This is step termination of translation. The final stage is termination when the ribosome reaches a stop codon in the mRNA. The first step, when a ribosome reaches a stop codon on mRNA, the A site of the ribosome accepts a protein called a release factor instead of RNA. The second step, the release factor hydrolyzes the bond between the tRNA in the P site and the last amino acid of the polypeptide chain. The polypeptide chain is thus freed from the ribosome. The third step, the two ribosomal subunits and the other components of the assembly dissociate. Translation. The final step in translation is termination. When the ribosome reaches a stop codon, there is no corresponding transfer RNA. Instead, a small protein called a release factor attaches to the stop codon. The release factor causes the whole complex to fall apart. Messenger RNA, the two ribosome subunits, the new polypeptide. The messenger RNA can be translated many times to produce many protein copies. Transcription. DNA is transcribed from a DNA template. In eukaryotes, the RNA transcript pre-mRNA is spliced and modified to produce mRNA which moves from the nucleus of the cytoplasm. Formation of initiation complex. After leaving the nucleus, mRNA attaches to the amino acid activation. Each amino acid attaches to its proper tRNA with the help of a specific enzyme and ATP. Translation: a succession of tRNAs add other amino acids to the polypeptide chain as the mRNA is moved through the ribosome one codon at a time. When completed, the polypeptide is released from the ribosome.